Good afternoon. So I will be presenting about the disease resistance in indigenous chicken. And my title of presentation will be the major histocompatibility complex genetic diversity of Kenyan indigenous chicken population based on microsatellite markers. Indigenous chicken are widely distributed under diverse geographical and ecological conditions. The geographically isolated indigenous chicken, normally called ecotypes, are subjected to variable climatic conditions. Such ecotypes are anticipated to possess unique combination of alleles, own genes, that may constitute adaptation to local environment. Kenyan indigenous chicken ecotypes are known for good adaptability to harsh scavenging conditions and poor nutrition, as well as tolerance to disease and parasites in their local habitats. These ecotypes may have evolved independently and become genetically diverged as a result of natural selection imposed by the local adaptation to climate, parasites, diseases, and nutrition in their local habitat. For this indigenous chicken ecotype to survive, this chicken must have a large plasticity in their immune system to be able to withstand a large number of immune challenges. This plasticity in this IC might be explained by the differences in the alleles of the genes in the major histocompatibility complex. The MHC is associated with immune response and disease resistance. Microsatellite markers, allele-0258, is a well-studied marker of MHC, and together with another marker called the MCW0371, have been known to explain different immune haplotypes. So also the marker allele-0258 has been used for several genetic diversity studies. However, in Kenyan situation, the relationship between the MHC and the indigenous chicken found in different ecosystems has not been studied in depth. So the objective of this study was to investigate the MHC markers, allele-0258, and MCW0371 marker of the different ecotypes of indigenous chicken, so that we can quantify the within and between population genetic differences. The ecotypes studied were 8 in number and were sampled in different regions of Kenya. As shown, we have the Lamu, Turkana, Kakamega, Siaya, Bomet, Narok, Taita Taveta, and West Pokot. For the methodology, 2 mature chickens were sampled per household, which was located at least 500 meters away. And in total, 768 birds were sampled. That is 96 samples per ecotype. Also, 1 bird per household was genotyped, so that the probability of sampling genetically related birds were reduced. All samples were collected from free-ranging indigenous chicken. Blood samples were drawn from the wing vein. For statistical analysis, both markers, the allele-0258 and the MCW0371, were examined individually and were also examined together. The genetic diversity was assessed by calculating the number of alleles per marker and per population, as well as observed and expected heterozygosity, plus overall population inbreeding, within population inbreeding, and within population inbreeding coefficients were calculated. This was achieved using the first one, the Alex software, which is used in estimating the observed and the effective number of alleles. Also, a population software, version 1.2.32, was used to estimate the allele frequency and the private allele identification. Another software called the FSTAT was used to calculate the inbreeding coefficients. Another software also called the structure software, version 2.34, was used for clustering. And this was run 100 times for each cluster K with a band of a minimum of 50,000 followed by 100,000 Markov chain Monte Carlo iteration. The structure harvester software, version 6.93, was used to analyze the output from the structure software, version 234, so that the optimal number of clusters could be identified. The number of clusters were identified using the ad hoc statistic change in K, based on the rate of change in the log probability between successive K values. These are the results which are obtained. In the 8 populations, that is the 8 ecotypes, 46 differently sized lay 0258 alleles were observed, with a size ranging from 194 to 550 base pairs. The 46 lay 25 alleles generated 229 genotypes, of which 35 were homozygous, whereas for marker MCW0371, a total of 10 alleles with a base size ranging from 198 to 207 were observed. In the table there, those are the number of alleles. You can see here, this is the observed number of alleles. This is the effective number of alleles, observed heterozygosity, and the expected heterozygosity. This one is the within population inbreeding. And if you can see for the late 258 marker, the number of observed alleles range from 20 to 27, the highest in the world. The nearest which can follow is 19 from Tanzania. The rest, there are 5 alleles, which means in terms of disease resistance, they are super. For marker MCW0371, only 10 alleles were recorded, and the number of observed alleles was very low. The maximum was 10. If you can see the distribution there from that figure, it varies from one ecotype to another. And the one with the bigger cycle shows that the frequency of that allele is higher compared to other alleles. Also, the results which were obtained was 3 haplotypes, that is BW3, B2, and B15 were observed. These were only 3 haplotypes which were observed and identified out of the 15 haplotypes recorded in the study. The remaining 13 haplotypes still remain at large. And out of the 3 identified haplotypes, haplotype B2 is known to confer moderate resistance to Marek disease. And the population which had the haplotype B2 was West Pokot ecotype. Also, after running out the cluster analysis, as you can see from this table, if we can use the marker MCW371, we cannot able to cluster our ecotypes. If you can see from the delta K, if you can see from the delta K here, the maximum is 1, 1, 1. So we cannot say the number of clusters is 7, or the number of clusters is 6, or the number of clusters, if you can follow this one, is 3. So the conclusion is that you cannot cluster indigenous chicken based on marker 0371. But if you can use marker lay 0258, if you can see from delta K here, you can see the value, this value, it's extremely high. And this one gives us the number of genetic clusters, if you can follow here, in terms of disease resistance. So we have 2 clusters. Also, if you can combine the 2 markers, the number of clusters in terms of genetic resistance, as it is shown here with the maximum value of 40, is 2. So the optimal number of clusters were identified to be 2, and this one is in agreement by study by Desta, who reported that the Kenyan population belong to 2 genetic groups. In conclusion is that Kenyan indigenous chicken host many alleles, and these MHC alleles play a significant role in terms of disease resistance. And in terms of cluster analysis based on the MHC markers, 2 genetically distinct groups can be formed. One is Lamu and the others. Thank you.